Matthew Mazzelzun, a geneticist.
And I'm Franklin Stahl. You may recognize us as a scientist who supported Watson and Crick's theory that DNA replication is semi-conservative. Now we have made a device that replicates our experiment within a shorter time frame and less work on your part.
Before we get into the specifics of our product and what it entailed, we are going to quickly explain DNA replication. DNA helicase unwinds the helices and separates the strands, making them templates. The enzyme primase attaches quickly to each parent strand to create a base for replication to begin. DNA polymerase creates complementary strands through base pairing, where adenine pairs to thymine and cytosine to guanine. Each pair consists of an original and new strand.
Complementary base pairing is important for translation because cRNA molecules contain anticodons which are complementary to the codons of the mRNA. cRNA molecules bind to a specific amino acid that corresponds to the anticodon.
There were many components to our experiment, but we are going to provide you the summarized version.
To begin, we decided that using an isotope of nitrogen to tag the parent cell would be the most effective method because nitrogen is present in all nucleotides. We changed nitrogen in the atoms of the parent strands to have an extra neutron in the nucleus, making it heavier.
We grew E. coli in two different solutions, one with nitrogen 14 and the other with nitrogen 15. After a few generations, we put E. coli with nitrogen 15 and the solution consisting of E. coli and nitrogen 14. From there, we observed that DNA replication could not be conservative because it was DNA strands with intermediate density, and it could not be dispersive because it was also not a single density.
At first, this may seem like a complicated procedure, but the results clearly show that each parent strand is used as a template to make a complementary strand. Semi-conservative replication is the most efficient method because unlike conservative and dispersive replication, it is less likely to have mutations. Despite the rapid speed of replication, it is very accurate. It ensures that the copies of DNA strands are identical due to the base pairing. If the pairs were either 100% conservative or dispersive due to the absence of a parent strand, the body would not know if there was a mistake in replication. DNA polymerase sometimes makes mistakes during replication, but because it is semi-conservative, it is able to find where a base was incorrectly matched through proofreading and correct the problem. PCR produces large quantities of a specific target sequence of DNA, and the process consists of using denaturation, heating to separate the strands, annealing, DNA primers attaching to opposite ends of the target sequence, and elongation. Taq DNA polymerase, which is heat-tolerant, copying the strands.
The inspiration for our research came when I told Stahl that we could possibly use density labels to test Watson and Crick's hypothesis. I had heard Professor Jacques Monod talk at Caltech about chemical bonds and enzyme synthesis, which gave me the idea of using elements as a way of measuring changes in density. I give many thanks to Professor Max Delbruck, who taught me the biological aspects I needed to know so that the necessary experiments could be carried out. It was the reason why I was introduced to Watson and Crick's works. Admittedly, I did not know about their DNA structure model until he gave me their papers. Through their research, we were able to use the information to corroborate their findings. After 2 years of unsuccessful attempts, we were able to design an experiment that distinguished between parent and daughter cells.
Our experiment provided additional evidence for the semi-conservative hypothesis, and we decided to make a device that could be used by middle school biology teachers, teaching students about whether DNA replication is conservative, semi-conservative, or dispersive.
Hopefully, students will be able to use their deductive skills to come to the same conclusion as they did after the experiment.
These devices cost $495 each, but many states and research companies are giving out grants to help fund the buying of them. We hope you have fun using our new invention and visit musclesonstahl.com to learn more about our product.